Before moving to the microbes in the environment result, you should know that what is the colony. The colony is the visible microbial growth on a solid nutrients medium, and it's made up of the same type of microbes. So the colony means same type of microbes. So like binary fusion, the binary fission is a kind of asexual reproduction. So this is the most common form of reproduction in prokaryotes such as bacteria, and it occurs in some single-celled eukaryotes like amoeba. So in binary fission, DNA replication and segregation occur simultaneously. And then colony forming unit. What is a colony forming unit? So a cell or cluster of two or more sister cells that are capable of multiplying into a microscopic colony of cells. So basically, the colony forming unit is the unit which we use to estimate a number of viable bacteria in a sample. Viable is defined as the ability to multiply through the binary fission under the controlled condition. So different bacterial species usually have different colony morphology. So the colony morphology means different size, different colors, and we will take our plates out from the incubator, and using the marker we will count the total number of the colonies and the total number of different colony types. Sometimes you cannot differentiate different type of colonies, but we will do our best. So, and then you need to, on your lab supplement, you need to write down the location of your samples, number of colonies, and number of types of colonies. And you have to record it, but however, we don't need it in our lab report, but you need to record your results. We will move to the results. So all the TAs grab the samples from different locations. As you can see, soil, leaves, door knob, bench top, face, fountain, and we will see the result and discuss about it. For example, in soil, you can see a lot of colonies grows all over the plate. Different colonies, you can see 3, even 4 different colonies, and we cannot count it because all over the plates there are colonies. So we can say that too many to count. For the leaves, you can see that you need to count the colony. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 colonies. Oh, surprisingly, in a bench top, no colony. So there are 2 possible reasons. The one reason is 99% of microbes are unculturable. What does it mean? It means that they cannot grow. Why? Because maybe the temperature for them was not good enough to grow, or maybe there are a lot of reasons that bacteria cannot grow. The temperature, the timing, or what can else? The nutrient, yes, because all of them are the TSY plates. So maybe some bacteria use some specific nutrients to grow, but some others may not grow on that nutrients. The other reason is we just clean the bench top. So when you just clean the bench top with the alcohol spray, so typically you kill the microbes, so the bacteria would not grow. This is the bench top. But this is the face. You can see that a lot of microbes grow when we grab the, get the microbes from our face. So it means that our face is not necessarily clean. So you can count the colony 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, and you need to count all of the colonies. For example, this one is 300 colonies, okay? And this is from shoes. So as you can see all over the plate, the bacteria grows. Too many to count. For benchtop, 0 colony. This is from fountain, 0 colony. This is when we put the microbes, when we put the plate for 30 minutes in our benchtop, no bacteria grows. But sometimes you can see that there are tons of bacteria grows even when we put our plates on a room, I mean that on an open air. So here is 0. Cell phone, we already cleaned our cell phone, so there's just 3 microbes. And this is my hands, which I already cleaned it with hand sanitizer, 0. Which I already washed my hands, 0. But this is hair. Hair is another location which is pretty dirty. So you can see that there are a lot of colonies grows when we grab a sample from hair. It's for sure more than 200. This is the doorknob, 1 colony. So you know that this is a different situation in the campus. Typically for doorknob or for bench top or for elevator button, we're supposed to see a lot of colony growth, but for this time, because all the place are sanitizing by the staff, so nothing grow. And this is the floor. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 colony. So what is the conclusion from these experiments? It means that colonies, microbes are everywhere. And you can see that different colonies grows on a plate. But the growth of microbes are dependent on some of the things. First is the temperature. Sometimes the bacteria grows on 28 degrees, sometimes they grow on 38 degrees, and sometimes even go to 60 degrees. So maybe the temperature that these 28 incubator was good for one kind of bacteria, but was not good for the other kind of bacteria. The other thing is, as I mentioned earlier, is about the nutrients. So some bacteria use some kind of nutrients which is not good for another bacteria to grow. So based on these different things, and also based on sanitizing somewhere in the lab or our hand, you can see that we can decrease the amount of colonies grows on the plate. So you do not write a lab report for this experiment. This experiment is something that you need to do yourself. It was a fun experiment. I'm sorry you missed that. But then we will move to the broth culture experiments.